Chapter 3, Section 2, Electron Transfer Reactions. As we discussed in Section 1, the catabolic reactions are there because, to give up excited electrons. There's going to be electron donors. But for catabolism to continue on and not get halted, 2 steps into, or 3 or 4 steps into glycolysis, we see that there has to be a pretty set flow of who's giving up to who is accepting. If this flow gets interrupted, well, metabolic process is shut down because it's going to, as we're going to see in this chapter, the enzymes are dependent upon these, redox reactions being able to go through, and anything that halts them halts the ability of the enzyme to take a substrate and turn it into a product. We've studied many things that we find in the cell that are involved in the cellular respiration, whether it's prokaryote, eukaryote, and we have calculated out, we've looked at what we refer to as the reduction potential. What is the potential for this, compound to accept an electron? What is the, what is the probability that the potential of this compound to give up an excited electron? So these redox reactions, we now can not only sit there and say, okay, the compound A is going to be oxidized, compound B is going to be reduced, but with these reduction potentials, we can clarify what is, again, I hate to say the potential, what is the probability compound A will give up an electron? How strongly does it want to give up the electron? What is the probability that compound B will take the electron? How strongly will it have an affinity for that electron? We discuss things in the terms of half reactions, so this way we can sit there and honestly say who's been oxidized. Here's equation 1, and then, or the first reaction, and then we talk about who just got reduced. That's the second reaction. So they come in reaction pairs. And we look here at what they refer to as the Redox Tower. The higher up they are, the greater the potential for these to be oxidized. Down lower here, the greater the potential to be reduced. And if you look at it, here's things that are going to give up an electron. Here's things down here are going to want to take that electron. So, glucose, okay, this is our source of chemical energy. We always talk about oxygen, the final electron acceptor. So the electrons that glucose give up will ultimately end at oxygen. They may be passed through, cytochrome B, C, A on their way, but boom, 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 ultimately that's where they'll be. And we look at here, when we talk glucose, oxygen, boom, there's the first part, here's the second. It's not oxygen as the final electron acceptor, it's going to be an inorganic, so this is an anaerobic. So we can sit there and we can discuss glucose plus plus oxygen versus glucose and ammonia, or nitrite, sorry. Who's got the better potential? Who's got the greatest potential for this to occur? Whereas what's going to give us the more energy, free energy? We always sit there and talk about who's the oxidized reactant, the donor, who's the reduced reactant, who's going to accept. So you have to keep straight: oxidized, reduce, reducing agent, oxidizing agent, the oxidized reactant, the reduced reactant. So that reduction potential, Allows us to sit there and quickly determine who's going to give up, who has the greater potential for accepting when we're looking at a reaction mix. And then you go through it, you compare. I mean, pyruvate and glucose. You know, these are organic compounds. -70. 4 electrons are going to be given up. You go through and here's different things that can be utilized throughout the cell. Some of them are going to want to give up, and some of them in certain cells are going to want to take. So we write these off as half reactions. Who got oxidized, who got reduced. It's a means for us to sit there and instead of writing A plus B, you know, equals A plus B minus blah, 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 and not understanding what's going on, who got the electron, where did it go, we write them out separately so that way we have something to talk about. Now, a lot of times, though, it gets a little more difficult because A and B are not directly interacting with each other. There has to be a go-between. Well, those go-betweens we refer to as electron carriers. Electron carriers are going to act as chaperones, okay? Their job is the electron movement, taking the excited electron, hence hydrogen, from A, transporting that excited electron / hydrogen to wherever B is in that cell. NAD will be reduced to NADH. Okay, H, right there is that excited electron. It's just bringing along a proton right now. They refer to NAD and NADH as a redox couple. Okay, NADH is a good donor. It's going to want to give that up. And NAD is a kind of a weak acceptor, which is why you need to have an enzyme there helping it along. It's not just going to pick up a stray electron, like, you know, on the previous slides when we were looking at that table and they listed in there iron, ferrous iron. Yes, iron will take up an electron that just happens to be coming by. NAD won't. So it needs an enzyme to help it out. Here's our friend, NAD, nicotine, amide, adenine dinucleotide. Dinucleotide, it's 2 nucleotides, modified nucleotides. So NAD, boom, boom, boom. See adenine up here, the nicotine amide. This is the cyclic ring. And right there, right there is where that electron will go. Along with it will come a second hydrogen that will go to another one. So 2 hydrogens. Here's the excited electrons. They displace that one. Boom. You'll also see in textbooks, primary literature, they'll refer to this as NADPH. The difference between NAD and NADPH is right here, this hydroxyl. If the hydroxyl here, it's NAD. If that's replaced with an inorganic phosphate, then it's NADPH. NAD, NADP, these are referred to as coenzymes, okay? Like I said, the enzyme that's in the process of, you know, taking a substrate and turning it into a product, and in the process is going to strip off an electron. NAD or NADP has to be there, has to be part of the active site, or whatever reaction that enzyme is, you know, catalyzing will not occur. And as we see it here, enzyme 1, right here, NAD has to be in there along with the substrate in the active site. In the process of being there, as the substrate is turned into a product, the enzyme's doing its cut, paste, twist, tweak, whatever it does. Excited electrons / hydrogens being pulled off, added to NAD to now reduce it to NADH. NADH, well, it'll take that electron to some other place, some other enzyme, some other protein, transfers it again, gets oxidized back to NAD, goes back to enzyme 1. And we just see this. The energy for enzyme 2 to cut, paste, twist, tweak its enzyme, or I'm sorry, its substrate, comes from that excited electron that NAD is delivering here. So now that it's released here, that electron / hydrogen is in a lower energy state.